Hello, guys. Good morning. Welcome to Nature's Blueprint. Uh, Today, I am going to discuss about bacterial genetics. As we have discussed earlier about binary fission in bacteria, in binary fission, the genetic material firstly divide, and after that, division of cytoplasm also take place, and the single bacterium divided into two daughter bacteria. Okay, so this type of transfer of genetic material from mother to offspring is known as vertical transmission. When we talk about horizontal transmission, here the genetic material is transferred from one bacterium to another of the same generation. Okay, so when the genetic material is transferred from one bacterium to another of the same generation, then three mode of transmission occur, and they are categorized as conjugation, transduction, and transformation. These are the example of horizontal transmission. Okay, so today we are going to discuss about bacterial conjugation. Uh, Conjugation. What is conjugation? Conjugation is the method of transfer of genetic material from one bacteria to another when they are placed in contact. Okay, this method was proposed by Liderberg and Tatum. In conjugation, you can say one bacterium as a donor and the other is recipient. The donor bacterium has a DNA sequence called fertility factor, or you can say F factor. So, the process of conjugation is discovered in E. coli, and here the donor having F factor have the ability to transfer it to another bacterium which is known as recipient, which doesn't have the F plasmid. Okay, so here we are going to discuss about the mechanism of bacterial conjugation. So bacterial conjugation involves four steps. First is pilus formation. Second one is physical contact between donor and the recipient cell. Third is transfer of F plasmid, and the fourth one is synthesis of complementary strand. So here in bacterial conjugation, two bacterium, one having donor F plasmid is known as donor, and the other which doesn't contain any plasmid is termed as recipient. So first of all, pilus formation take place. The donor cell form a sex pilus. The donor cell which have F plasmid have the ability to form sex pilus and begin contact with an F minus recipient cell. Physical contact between donor and the recipient cell. The pilus start to form conjugation tube, which enable direct contact between the donor and the recipient cell. After that, the F factor open at the origin. This F factor, F1 F plasmid open at the site of origin of replication, and the 5 prime end enters the recipient cell. 5 prime end start enter into the recipient cell. After the whole transmission of single strand of plasmid in the recipient cell, both the donor and the recipient cell start to synthesize complementary strand. Okay, so now the recipient cell have a single copy of F plasmid, and the donor cell also have single copy of plasmid. So both started synthesizing complementary strand, and now the recipient also can act as a donor cell. Okay, so this is all about conjugation in bacteria. If you have any doubt, you can ask me in comment section. Thank you for watching this video and wait for the next video. Thank you so much.